5 is more spicy.
So we need to talk about just a couple things today. We'll talk about acidity, and then we'll move into water specifically, and then we'll talk about carbohydrates. I have to find my clicker. So, you've heard of acidity. We talked about it a little bit back in 7th grade when we talked about acid rain, acid precipitation. And acidity is basically a property of a solution that you can measure. Just like you can measure the temperature of a solution or the amount of oxygen dissolved. Acidity is another measure of a substance. And water, we're going to talk about in a few minutes, is a polar molecule, which means the different parts of the water molecule have a charge to them, okay, positive and negative charge. And so sometimes when you have water, it dissociates, and the hydrogen separates from the hydroxide, the H from the OH, and in a neutral solution, that's in equal amounts. There's equal amounts of hydrogen ions and equal amounts of hydroxide ions in pure water. We call that a neutral solution. We would say it has a pH of 7. We'll talk about pH in a minute. In some cases, there is a higher concentration of hydrogen ions than there are hydroxide ions, and that's an acidic solution. The opposite of an acidic solution is called a basic or an alkaline solution, and that's the opposite case, when you have a higher concentration of hydroxide than you do hydrogen ions. And so these are the properties that we measure the acidity or the alkalinity of a solution. And so you guys mentioned it earlier, you probably, maybe you've done this if you have a pool. You have to measure the pH of your pool. Does anyone have to do that at home?
No.
Do you know what the number is? What are you looking for in the pH of your pool? Do you know, Gabe?
I know there's some kind. It's been a while since I've done it. It's been like since August, so I kind of forget. But there's like a scale that you're supposed to.
Yeah, you compare the color.
Yeah.
And maybe it doesn't have a number with it. You're just trying to match up the color.
Yeah, that's our
So
scale.
those colors actually have a number associated with them that describes the acidity of that solution. Okay. We'll talk about that. If you take an acid and a base and mix them together, they neutralize each other. And an example of that is if you've ever had heartburn. Okay. Has anyone had heartburn in the past? So when you have heartburn, acids from your stomach has hydrochloric acid in it. And when that acid creeps up into your esophagus sometimes, it damages it, it burns, and it hurts. And so what do you take in those cases? So yeah, Tums, something like that, Alka-Seltzer. Those are basic compounds. Those are chalky substances, and when you chew them up and swallow them, they neutralize the acid that's in your esophagus, and they make it feel better. So they work on this principle of acids and bases neutralizing each other. All right. So the scale we use when we're measuring acidity is called the pH scale. And the pH scale, it comes from the words potential hydrogen, because that's what acidity is, how many hydrogen ions there are. It was developed in 1909, and the thing you guys were confused about and you're asking questions is a low pH number indicates greater acidity. So the scale itself goes from 0 to 14. In the middle is 7, obviously. That's a neutral substance. So pure water has a pH of 7. Lower numbers are more acidic. Higher numbers are more basic. So each step you go, each whole number is actually a 10-fold increase. It's an exponential scale. So if you go from a pH of 6 to 5, the 5 is 10 times more acidic than 6. And 4 is 100 times more acidic than 6. So it's an exponential scale. Things that are at the extremes are things that can be very dangerous. So battery acid is corrosive, can burn your eyes and your skin. It's a pH of about 0.5, so that's really low. Our stomach acids are around 2 to 3. Foods that we might be familiar with, lemon juice is very acidic, a pH of 2.4. Apple juice, orange juice, vinegar, those are all acidic foods. At the opposite side, something that's extremely basic is also dangerous, is also corrosive, can also damage your skin and be dangerous to your health. Things like bleach and ammonia are at the basic end of the scale. Soap is slightly basic. So seawater is slightly basic. What's that?
So like, if bleach is more basic, does that mean it's like healthier than water?
Yeah, like.
Is it what?
Healthier than pure water?
No.
No, it's an
That's-
extreme.
No,
Pure water is
closer
7.
to 70. Yeah.
So-
Yeah, 7 is neutral.
Lemon juice can be like more dangerous than hand soap.
I mean, neither are dangerous.
But like, it's like.
It's more acidic, yes. Because hand soap is basic. It's on the opposite side from lemon
What
juice.
does it mean it's dangerous too?
It doesn't have to do with danger, really. Extremes, just like extreme temperatures can be dangerous, extreme pHs can be dangerous, but also the central areas are not. Luke?
So like in the middle is like the most like the not like the neutral ones. And
7
then
is neutral,
if you go
yes.
to the right or go to the left, that's the more you have.
More basic to the right in this diagram, more acidic to the left.
What do you mean by basic?
So basic.
That's the opposite of acidic. It just means,
So you read it from right to left?
you don't read it anyway, it's just a scale.
The
It is
middle
what it
is, the middle is like
Just
zero.
don't-
It's
7 is pure water, completely neutral. So we use indicators sometimes to try to measure pH. And there's a whole bunch of different indicators that you can use. An indicator is just a substance that changes color to measure something for you. So I have some examples here. So this is phenolphthalein. This is a clear solution. It's this first one. It starts off clear, but it changes color. Okay, so you see on the diagram it says starting color is clear for phenolphthalein. And then when it's basic, it turns red. When it's acidic, it stays clear. So if I make this solution more basic, it'll change color. So what could I add that would make it basic? Soap would make it a little more basic, maybe not enough. Yeah, if I added bleach or ammonia, those are at the extreme end on the basic scale. This is just like ammonia by the grocery store or whatever. And so if I add that, you should be able to see a color change.
Magic.
Oh, that's
Wow.
purple.
That's pink.
Yeah, that's like pinkish purple. Sometimes it will go back. So as the indicator molecules interact with the hydroxide ions that make this basic, it will sometimes go back until I put more ammonia in it. Sometimes it won't. And then you can change the color again. So what can I do to change this back to clear?
Add
Make
more
it
water.
acidic.
Oh, add lemon juice.
Add something acidic. So if I take some vinegar,
Milk.
if I take some vinegar and add it, I should change the pH back to more acidic and it should be clear.
Magic.
Whoa. I missed it.
So this is an indicator.
You said you're a magic.
Use a box.
Another indicator, bromothymol blue.
Yeah, that's so.
And, this is bromothymol blue. So this is also a pH indicator. It changes color in sort of the neutral range. So when it's basic, a pH above 7.6, it's blue. When it gets to be neutral, it's green. When it gets to be acidic, then it's yellow. So that's obviously, what is this? What pH?
7.6.
Above, somewhere above that. We don't know exactly, it just is somewhere above that. So if I pour some of this bromothymol blue, what could I do to change the color?
Water.
Add more acid.
Well, water would make it slightly more.
Bleach.
Yes.
Bleach.
Bleach
No,
is a
no.
base.
Vinegar.
Vinegar is an acid. I could add that. Or something else that's kind of neat. So when you exhale,
Carbon dioxide.
carbon dioxide from your lungs is, when it mixes with water, forms carbonic acid, and that's a mild acid. It sometimes takes a while, but if you exhale into this for long enough, it will change. I'll see if I
Don't
can-
suck it in.
Yeah, I know, that's bad. It's
Yeah.
toxic.
Will you die?
Maybe.
Careful.
What I did
What
to
if
my
it just
chocolate
changes
milk
in
this
the
morning.
Sometimes exactly like sucking.
Oh, wait, whoa, what?
All right, so we're about neutral.
Wow.
It's
It's cool.
green.
It's green.
I can't see it.
It's green. it gonna feel yellow right now? No.
Yo.
Stop playing.
What?
Stop. You play too much, Mr. Jones. You play too much.
I don't think it's changing.
It's changing.
That's a little yellower than
It's
before.
yeah.
What's
It's
the
more.
what's the
It's a
song?
little harder.
What's the song?
Now it's like yellow.
No, it's changing.
It's not it's yellow green.
I see it now. This is
Whoa! Mr. Jones, you got magic.
Can you keep
We
on?
get light.
Would it eventually turn red?
No, it goes to yellow and that's
Does
it.
it go back? Oh my God.
Wait,
It
looks like you're.
I went blue to green so fast.
Wait, would it go back?
Okay, let's sort it out.
Probably. So if you drink it.
It's yellow.
Yellow now?
No,
It's
no.
definitely
You're
yellow.
like passed out, so.
Get out of
What
this.
are you doing in my
Alright, well, what's gonna happen if I add ammonia? Blue.
Azul.
See this, I'm like red.
Yeah, yo.
Oh, come on, you won't call that for
Yo.
Try adding here. Oh, vinegar involved.
Yeah, vinegar.
Right.
Let's
Yellow.
Here we go.
Whoa.
That's like apple. That's like apple juice.
Alright, so that's
Tasty liquid.
that's bromothymol blue.
Boy, you've been
Another-
a long time.
Common,
Red litmus paper.
I drink.
another type of
I drink water.
indicator is litmus paper. So, what do you have? Vinegar. So,
Boy, what are you doing with yourself?
That smells like
so there's a couple types of litmus paper. So this is red litmus paper. Red litmus paper starts off red. When the pH goes above 8, when it becomes basic, it turns blue. So let me,
Is basic always blue?
Well, you basic.
yeah, these, well, not always, but
Usually?
yeah.
You basic.
You basically are.
Basic.
So if I have some ammonia, if I put the red litmus paper in ammonia, turns bluish purple. The other thing you can have is blue litmus paper, which starts off blue when it's basic, and if you put it in an acid,
no way. It turns red.
turns red.
Oh, sponges.
That's wet paper. Oh my god. We're eating acid.
And then the other type of paper,
White, yellow.
this is called P hydron paper, and this changes. This is probably similar to what you use in a pool. This gives you a broad range. This will turn color, and then you match it up to the scale that's on the bottle, and you can figure out this is actually above. Some have another color beyond this green, so this would be a pH somewhere above 10.
15.
Or if you put it in a
That's pretty bad.
if you put it in an acid, it'll turn one of the other colors. And they make these in a whole range of pHs. Like some will, this only tells you like every 2. Some will tell you if it's 1, 2, 3, all the way up to 14. Some are in a narrow range. They'll tell you if it's like 6.2, 6.4. Just depends on what type of paper you have.
No, yeah, you did that.
Yeah.
Yeah, so this one red would tell you that's around pH of 2.
Wow.
Okay. Maybe it's not quite 2, 2 to 3. So these are all indicators that are used to measure the pH of a substance. You also could have pH meters. You just have a little digital meter you put in the substance. It'll tell you what its pH is.
She should really start.
pH sensors. All right, so we saw that. Litmus paper. Oh yeah, this is like a more, this one's a little more detailed P hydron paper where it tells you 0 each whole number, all the way up to 13. It just comes in different types. All right, so we need to talk about ice, about water and some of the unique properties of water. Water has some chemical properties that make it pretty unique. And without these properties, probably we wouldn't have life on Earth. So we know, you obviously know water is H2O. So 2 hydrogen atoms covalently bonded to an oxygen atom. So an oxygen has 6 valence electrons. So it's looking for 2 more to get to a full level of 8. Hydrogen has 1 electron. And actually for hydrogen, 2 is a full outer shell because it's very small. So if each hydrogen atom shares one of its electrons with the oxygen, then the oxygen has 8 electrons total. Each hydrogen has 2 total. So they all have a full outer shell, and therefore it forms a stable compound. But the way the electrons are shared, so these, what type of bonds are these? Covalent bonds. But there's another term we sometimes use. So if I have, let's imagine this is an electron. Stand up. Oh, no. This is an electron. Okay? Let's say I'm oxygen. Gaetano's a hydrogen. Okay, we are sharing this. We are sharing this electron. We have a covalent bond. Now, if we're sharing it equally, all right, that's a regular, we call that a non-polar covalent bond. But if I can pull on that, come with me. If I pull on that stronger and the electron is closer to me, I take on a partially negative charge. And Gaetano, because the electron's closer to me, takes on a positive charge. Aiden, come here. If we are a water molecule together, the 3 of us,
Take a picture.
so if the electrons, wait, we gotta have everyone see us hydrogen. All right, so in a water molecule, the oxygen is pulling in these electrons closer, well, not so close. So the oxygen takes on a negative charge because the electrons are closer to me. The hydrogens take on a positive charge because the electron's farther away from them. Thank you, hydrogen molecules. That's so sweet. So what this means chemically is that if we imagine a water molecule, the oxygen portion has a slightly negative charge. The 2 hydrogen portions each have a slightly positive charge. What this means is that water molecules are attracted to each other. That the hydrogen is attracted to the oxygen of another water molecule. And that's what gives water its cohesive properties. If you remember back in 7th grade, we put those drops of water on the penny, and it could hold like 40 drops of water. The reason is because the water molecules are slightly sticky with each other due to these positive and negative charges. That type of bond we were just illustrating is called a polar covalent bond. We're still sharing electron, but we're not sharing it equally. One molecule is attracting it closer, giving it a little bit of a positive and negative charge.
What if an oxygen molecule had 4 hydrogen molecules connected to it?
It doesn't, because the electron configuration wouldn't require that. Because once they form a stable outer layer, they don't really interact with other elements. So these properties of water are really important. So first of all, the basics are that 2 hydrogen atoms are joined to an oxygen atom by single polar covalent bonds. Polar covalent bonds. Polar means they have a charge on one side. Covalent means they're sharing the electron. And that means 2 separate water molecules will form what we call hydrogen bonds with each other. These are relatively weak bonds. They can be broken pretty easily, but they still are there. There's still an attraction between water molecules. So this results in water molecules having a cohesive force. They want to stay together. They'll pile up on top of that penny. You could fill up your glass of water slightly above the rim because the water molecules are holding on to each other. It also results in surface tension. A water strider insect can walk over the surface of the water because the force of it pushing down is not stronger than the cohesive force between the water molecules, and it doesn't break through. Water has a high, if you did that Penny Lab thing with alcohol, it would not hold nearly as many drops because the alcohol doesn't have these cohesive forces. You might probably remember from earth science that water has a high specific heat. Has a specific heat of 1 calorie per gram degree Celsius. It takes a lot of energy to change the temperature of water. So what are climate conditions like near large bodies of water compared to areas that are just land?
Like warm and tropical.
They're, well, not necessarily tropical, that depends on the latitude, but they're moderate. There's not as many extremes. Like if you live the same latitude but you're right on the ocean versus in land, an area surrounded by all land, it's going to get hotter over the land and colder over the land. The oceans moderate that temperature. It also allows us to have evaporative cooling. When water evaporates, it takes with it energy. That's why when we sweat and the sweat evaporates, it cools us down. Water is an extremely versatile solvent. It can dissolve many, many substances. Not all, but many. And so these properties of water that are unique are important to the biochemistry of living things. Let's take a break. Slide. So what we're going to start to do now is talk about different classes of molecules that are important in biochemistry. So first of all, if you remember, what does organic mean?
It's alive.
No.
No, not in biochemistry.
It is
Made in earth.
less acid.
No.
Like it's natural.
Okay, so maybe, did we go over this or not?
Yeah.
Okay.
Yeah, it's made by living organisms.
So organic, you have to sort of separate what you hear like in everyday language. Like organic in terms of biochemistry has nothing to do with like herbicides or pesticides or foods. In chemistry, organic means a compound has carbon and hydrogen, both. So these are molecules typically that are made by living organisms. And carbon is a key portion of that. Carbon is the most abundant element in living things. And carbon, do you remember how many electrons it has in its outer shell, carbon?
Two.
8.
You just guessed.
7.
It has 4. The only number I
knew
would
it.
say.
I said 4.
Has 4 electrons in the outer shell, which means that it can form 4 separate bonds with other atoms. That gives it a wide variety of shapes and configurations and so forth. And that results in a huge variety of molecules containing carbon. So if we think about a carbon-based molecule, it can vary, they can vary one to the other in terms of how long the carbon chain is. You know, 2 carbons versus 3 in ethane versus propane. The position of double bonds changes. Here we have 2 carbon atoms that are bonded to each other with a double bond rather than these single bonds. It could be here or it could be here. And that changes the molecule. Sometimes the carbon skeleton of the molecule is just linked in a straight line. Other times it branches. Sometimes it forms this ring shape. Sometimes it's a hexagon, sometimes it's a pentagon. It depends on how many different carbon atoms are in the ring. So there's a variety of shapes and configurations for carbon-based molecules. So in biochemistry, we're going to talk about these different classes of biomolecules. Carbohydrates, lipids, proteins, nucleic acids, and then these other high energy compounds. And so we're going to start today by talking about carbohydrates. We'll get into some fair amount of detail. And so carbohydrates include polysaccharides, disaccharides, and monosaccharides. We'll talk about each of those. Now you know, I'm sure from 7th grade, from health class, about carbohydrates in our diet. What are carbohydrates used for in our diet?
Short-term energy?
Yeah, energy. They're an excellent source of energy. In fact, that should make sense because what chemical reaction that happens in cells uses a carbohydrate.
ATP.
ATP is not a respiration. Because what is the carbohydrate it uses, Francesca? Glucose, and that's a carbohydrate. So carbohydrates provide energy. They have 4 calories per gram.
We did that in health.
Yeah. Also, carbohydrates form structural parts of cells that give cells their shape and their size. A lot of times when we think about carbohydrates, we're thinking about starches and sugars. And the name itself, carbohydrate, the carbo means carbon, hydrate means water. And that's what carbohydrates are. They're just basically carbon and then hydrogen and oxygen in a 2-to-1 ratio. Oftentimes carbohydrates have the suffix OSE at the end of the name. Not always, but often. So glucose, sucrose, maltose, fructose, cellulose, these are all carbohydrates. The OSE ending clues you into that. And they're made of just 3 things, carbon, hydrogen, and oxygen. So these are examples of some carbohydrates. We'll talk about each group one at a time. The simplest group are the monosaccharides. What does mono mean? One. So these are the simplest. These are like the building block carbohydrates. They're made of just one ring or one set of carbon and hydrogen and oxygen. They're the building blocks. We call them the monomers. So these molecules are that are long chains are polymers made of these simple units called monomers. The basic molecular formula for a monosaccharide is C6H12O6. If we're drawing them, we often draw it as a hexagon, each angle representing one of the carbons. So here are 3 monosaccharides, glucose, fructose, and galactose. They actually all are the same formula. They're all C6H12O6. But the arrangement of those atoms varies from one to the other. We call them isomers. Glucose, Francesca just mentioned. It's a very important sugar in living organisms. It's used in cellular respiration. Basically, any carbohydrate that we eat will be digested into glucose in our digestive system, absorbed into our blood, and then our cells can actually use it. Fructose is a plant-based sugar. It includes honey has fructose, plant nectar that's used to attract pollinators. Fruits have fructose in them. It's a very sweet sugar in terms of taste, and that's why fructose is used for what? Yeah, sweetening. Almost any food product you buy that's like mass produced is not sweetened with sugar. Sugar is sucrose, but it's sweetened with corn syrup, high fructose corn syrup, which is mainly fructose. It's very sweet, so it's cost effective, it's efficient to use in cooking. Another monosaccharide is galactose. Okay, galactose is a sugar that moves through the cell membrane very quickly. It's often one of the components of these huge polysaccharides. So since all of these have the formula C6H12O6, how are they different from each other? Well, if you look at the actual shape of the molecules, there are some differences. You have fructose, which has 5 carbons in the ring, whereas the others, glucose and lactose, have 6 carbons in the ring. The position of some of these hydroxide groups is different. Some are sort of pointing down, others are pointing up. But those small changes may not seem like much, but they actually change the behavior, change the functioning of that molecule. If we draw them here as chains, you see glucose and galactose are stereoisomers. It means just something is like mirror image switched, like the hydrogen and hydroxide here in the middle of the chain, whereas fructose has a different sort of arrangement of those atoms. If we take a couple monosaccharides and link them together, we form a disaccharide. A disaccharide is a double sugar. formula for a disaccharide is C12H22O11.
Wow. What the heck is malted milk?
You ever have a malted milkshake?
That is so good.
Or you ever have Whoppers, the candy?
Yes.
Oh yeah.
Malted milk balls?
Oh
They're
yeah.
made with maltose sugar. That was what gives them that flavor.
Is it like just porous sugar?
No, but that's due to how it's used. The sugar is used to sweeten and they make it that. But the porousness is not a property of maltose. So a disaccharide we would draw like this. It's two rings of monosaccharides linked together with an oxygen atom. Some examples, maltose. This is produced in seeds when they germinate. A seed inside of it has stored up energy in the starch. And when that starch starts to be digested as the seed germinates, it forms maltose, a sugar. It's also malted beverages like beer and other malt beverages are made with malted barley. They allow the barley seeds to start germinating so that the starch turns into maltose sugar, and then they stop them and they use them to brew beer by fermenting the sugars from that fermented or from that malted barley. Lactose, we talked about a little bit the other day. It's a sugar. It's a disaccharide sugar. So in order for us to use it, it has to be broken down. It's found in milk and dairy products. And then finally, sucrose. That's what we think of as sugar. Table sugar is actually a disaccharide. It's sucrose. Okay. So this is maltose, this is lactose, this is sucrose. And they have they're each different monosaccharides joined together. But they're all these disaccharides. You'll notice they all have an oxygen in the middle, which links them together. We'll talk about why that's the case in a couple minutes. But
So there's two sugars in sugar.
there's two monosaccharides in the disaccharide sugar. Yes.
Wait, why isn't it O12 and O11?
What's that?
Disaccharides.
Because so why isn't it double of the monosaccharide? Yeah, that's a good question, and I'll show you in a minute. Because when you join them together, you lose something. So how many hydrogens do you lose when you join them together?
1, 4, 2.
How many oxygen do you lose?
1.
So what do you think the byproduct is? Water. So when you link them together, you remove a water. 2 hydrogens, 1 oxygen. That's why this is not 24 and 12. Good question. So the last type of carbohydrate we'll talk about are polysaccharides. So poly means what? Many. These are often very, very large molecules. Many monosaccharides all linked together. So they're a large, what we call polymer. So we don't usually draw a huge ring. We could draw hundreds of these if we wanted to represent the monosaccharides. They can be 200 they can be thousands of monosaccharides linked together. Many of these are the structural carbohydrates that make up the shape of living things. So starch is an example of a polysaccharide. Starch is what plants use to store up energy. It's a polymer of glucose. So if you have a bunch of glucose molecules all linked together, that's what forms starch. We eat lots of foods. Our plant-based foods have a lot of starch in them. Breads, pasta, anything made with flour is rich in starch. Potatoes, because that's stored up energy from that plant. Cellulose, which you find in a plant cell wall, that's a carbohydrate. That's a polysaccharide. It forms the cell wall of plants. Chitin is what makes the exoskeleton of insects, and it makes up the cell wall of fungi. That's another carbohydrate, another polysaccharide. Another interesting one is glycogen. Glycogen is how animals store starch, or store energy. So for example, right now in your liver, you have a store of glycogen. And if you don't eat for a while and you need more glucose into your blood, your body mobilizes this glycogen to break down into sugar to go into your blood. Your muscles also have glycogen because when you're using your muscles, if they exhaust the glucose that's in your blood, they start to get the glucose from this glycogen stored in your muscles. If you exhaust that, then you're out of energy. If you do really long endurance athletics, like if you run a marathon, and not like 2 or 3 miles, if you run a marathon, you completely exhaust all of the stores of energy you have in your muscles, and so running a marathon, you have to eat while you're running. You have to take gels, or you have to
eat while they're running.
yeah, usually in form of gels or
Like that.
Gatorade, sports drinks. Some people eat like a banana while they're running.
My mom was the
Yeah, and so that allows you to get glucose because in 26.2 miles, you have completely used up all your stored glycogen. And so these are some examples, cellulose and starch and glycogen here. All right, the last 2 things to talk about is how these molecules get put together and broken down. So dehydration synthesis, let's just think about the words itself. So dehydration means what?
No water.
Removing water. Synthesis, this is really important to know what synthesis is.
Synthesis.
What does it mean?
Synthesizer.
Yes, well, a synthesizer
It does stuff.
makes sounds. Photosynthesis, light is used to
Make
make something. It means making something. It means building up a large molecule from smaller molecules. And so this is how these monomers, these monosaccharides, get linked together to form disaccharides. And get linked together to form polysaccharides through this chemical process called dehydration synthesis. So if we look, it'll make sense. It looks confusing probably with all these molecules and atoms and so forth, but it should make sense. We have 2 monosaccharides. There are 2 rings of carbohydrates. Okay? So these are monosaccharides. To link them together in dehydration synthesis, the hydroxide on one side gets kind of stripped off. The hydrogen from the other side gets stripped off. And we have a water molecule. And then this remaining oxygen just gets linked. So this bond gets linked to this oxygen, and then they're joined together. So we took that, the water was released, and now this is a disaccharide. So that's how they're produced. The opposite of that is hydrolysis. Hydro meaning water. Lysis means to break up. And so that's just the opposite of that. We need to add a water molecule. So here, what is this? It's a type of, I don't need the specific name. It has two rings.
Oh, man.
It's a disaccharide. What are these?
Monosaccharides.
Those are monosaccharides. So how do we split a disaccharide into 2 monosaccharides? It's the same process. So here, they're linked up at this oxygen. We add a water, okay, and the oxygen and hydrogen get added into one side. The hydrogen gets added to the other, and we split it into 2. So sucrose, table sugar, is made of glucose and fructose. This enzyme sucrase allows that to happen. So we need to add water to split a disaccharide. We need to release a water to join a disaccharide. It's the same process, it's just the opposite. So this is an example of digestion, breaking a larger molecule into 2 smaller molecules. Here's just another way to show it. We have glucose and fructose. We get rid of this hydroxide and this hydrogen by turning it into water, and we link these 2 up by the oxygen. Well, we do the opposite down here. We have them, a disaccharide, they're linked up by this oxygen. Okay. Add a water. Okay, the hydroxide goes to one side, the hydrogen goes to the other, and now we have 2 monosaccharides. So they're opposite processes. Any questions about carbohydrates?